Good evening, viewer. Welcome to our Glory Globe TV. Here's a breaking and most amazing news too for our viewers. Researchers at the Weizmann Institute of Science in Israel have identified a yeast that could be used to prevent invasive candidiasis, a major cause of death in hospitalized and immunocompromised patients. The study, published in the Journal of Experimental Medicine, shows that the novel yeast lives harmlessly in the intestines of mice and humans and can displace the yeast responsible for candidiasis, Candida albicans. Millions of microbial species live within or on the human body, many of them being harmless or even beneficial to human health. The microscopy is C. albicans is commonly found in the intestines and other mucosal surfaces of the body and is usually benign, though occasionally it may overgrow and cause superficial infections commonly known as thrush. Under certain circumstances, however, the yeast may penetrate the intestinal barrier and systematically infect the blood or internal organs. The dangerous conditions known as invasive candidiasis is commonly seen in healthcare environments, particularly in immunocompromised patients with mortality rates up to 25%. Studying yeast infections in laboratory mice, Stephen Young and colleagues at the Weizmann Institute discovered that some of their mice carried a novel species of yeast that prevented the animals from being infected with C. albicans. The new species, which the researchers named Kazakhstania weizmannii, is closely related to yeast associated with sourdough production and appears to live innocuously in the intestines of mice even when the animals are immunosuppressed. Researchers found that the K. weizmannii can outcompete C. albicans for its place within the gut, reducing the population of C. albicans in mouse intestines. Moreover, while C. albicans can cross the intestinal barrier and spread to other organs, in immunosuppressed mice, the presence of K. weizmannii on the animals' drinking water significantly delayed the onset of invasive candidiasis. Notable, Zhang and colleagues also identified K. weizmannii and other similar species in human gut samples. Their preliminary data suggests that the presence of K. weizmannii was mutually exclusive with the presence of Candida species, suggesting that the two species might also compete with each other in human intestines. By virtue of its ability to successfully compete with C. albicans in the murine gut, K. weizmannii lowered the C. albicans burden and mitigated candidiasis development in immunosuppressed animals, Zhang says. This competition between Kazakhstania and Candida species could have potentially therapeutic value for the management of C. albicans mediated disease. Behind the report, Glory Globe TV, Delhi, Bharat.